Today we discuss about very interesting topic. Next one, yeast cell. Look at here. This is the yeast cell. The yeasts are unicellular, having 0.1 to 0. Mu meters in thickness. This is the original pic of yeast cell. You can observe this is the yeast cell. Look at here. Yeast cell example Saccharomyces cerevisiae. This is the best example of yeast cell. The yeasts are unicellular. Uni means only one. Okay, having 0.1 to 0.2 mu meters in thickness. This is the thickness. Thickness, it is very, very important. 0.1 to 0.2 mu meters thickness. These are mainly composed of polysaccharides such as glucons, mannans, and chitins. Polysaccharides. What is meant by polysaccharides? Poly means many, saccharides means sugar. Okay, that is called polysaccharides, such as glucons. Glucons means glucose. Next, mannans. Next one is chitins. Okay. Next one is chitins. Chitin is an important component of the cell wall and septum. This is also example of yeast Saccharomyces cerevisiae. Cell wall made up of a chitin is an important component of cell wall. Okay, three chitin synthesis it can be made up of a cell wall. Okay, CHS1P, CHS2P, CHS3P have the same polymerizing activity but deposit chitin at different times and at different locations during of the cell cycle. Okay, this is the very important. These are mainly composed of polysaccharides. Poly means many, saccharides means sugar. Okay, next such as glucons, mannose, and chitins made up of a yeast cell wall. Next one, the plasma membrane in yeast cell is composed of a phospholipids and lipoproteins. Okay, these two are very important. Yeast cell composed of a phospholipids. First one is phospholipids, second one is lipoproteins. What is meant by phospholipids? Phospholipids main function. Phospholipid synthesis in yeast is a complex process that is regulated by genetic and biochemical mechanisms. Okay, this is the regulation of phospholipid synthesis. Next one, lipoproteins. Okay, second one is the plasma membrane in a yeast cell is composed of a phospholipids and lipoproteins. It is made up of a lipids and proteins. Yeast cell was first discovered by Dutch scientist Antony van Leeuwenhoek. Look at here. He is the Dutch scientist Antony van Leeuwenhoek. Okay. Who discovered yeast cell? This is the question. Antony van Leeuwenhoek. In 1680, Dutchman Antony van Leeuwenhoek observed yeast under a microscope for the first time. Next, yeast cell structure. It's very important. Yeast cell structure. Look at here. This is the bud. This is the yeast cell. I explained by one organelles. Look at here. This is the cell wall. This is the bud. This is actin cable. Next, this is called secretory vesicles. Look at here. This is the fat globule. Next one, Golgi complex. Next, this is called vacuole. Next, endoplasmic reticulum. This is mitochondria. This is cell wall. This is cell membrane. Okay. First, I explained by bud. What is mean by budding? The most common mode of vegetative growth in yeast is asexual reproduction by budding, where a small bud, where a small bud means also known as a bleb or daughter cell. Daughter cell, it is called small bud, is formed on the parent cell. This is the parent cell. Look at here. This is the parent cell. This is the daughter cell. This is called bud. This is the parent cell. Okay. The nucleus of parent cell splits into a daughter nucleus and migrates into the daughter cell. The nucleus transfer into the daughter cell. Okay. First parent cell in a nucleus splits into more daughter nucleus. This is the budding. Look at here. This is the nucleus. This is vacuole. This is yeast cell. Okay. First step, the nucleus splits. After this nucleus dividing in two nucleuses. Okay. One nucleus moves into a bud. This is the parent cell. This is the daughter cell. This nucleus transfer into the bud or baby cell. Okay. Next, this is the square. Square means divided into the two cells. This is the daughter cell. This is the mother cell or parent cell. Look at here. This is the parent yeast. This is the daughter yeast. This nucleus transfer into the daughter cell. Okay. This is called cytoplasm. Already you know. Daughter yeast and parent yeast separation. Look at here. Separated into a two nucleuses, two cells. This is called budding. Budding, it's very, very important. Okay. Next. This is the cell. Already I explained budding. Okay. This is the first yeast cell, haploid stage. Next, this haploid means alpha cells. Alpha cells divided into the scoma. Means after mating, mitosis process. Mitosis means cell division starting stage. That is called mitosis. Okay. Next, the cell division completed, the cells divided into two cells. This is the A cell, this is the A cell. After mating, A slash alpha cell means this is the alpha cell, this is the A cell. Okay, both cells are mating. What happened? This is the parent cell, this is the daughter cell. After mitosis process, it will be completed. Next, form into a two cells. This is the A alpha cell. This is the another A alpha cells. So how many cells is there? Two cells. This is called diploid. Di means two. Diploid means two cells formation. It will be done. Next, this is the diploid. But both are conjugated with the cell. But in this process, cells, it will be divided into two cells, separated of a two cells, a slash alpha cell. This is also called a slash alpha cell. This is the parent cell, this is the daughter cell. Next step, Mitosis process, it will be completed. Next step, meiosis. This is called meiosis. Meiosis means cell division process, multiplication process. Multiplication means two cells divided into four cells, four cells divided into eight cells. That is called multiplication. This process means meiosis. This is called sporulation or meiosis. How many haploid spores it's there? One, two, three, four. Four cells. Okay, this is the haploid spores. Next step, germination. After germination, how many cells is there? Four cells. Okay, this is the daughter cells, this is the parent cells. Okay, how many cells is there? Total four. This is called budding. Budding process, it's done. Okay, look at here, this is called budding. Next